The accidental plague. My name is Dar, Elias Thorne, and I never meant to become the harbinger of doom. It started innocently enough. In the sterile confines of my basement lab in a quiet suburb of Boston. I was a bioengineer by trade, tinkering with synthetic biology as a hobby after my day job at a pharmaceutical company grew stale. The world was already on edge in 2047, climate change ravaging coasts. Pandemics a fresh memory, but I thought I could make a difference. My project? Engineering a microbe that could break down plastic waste in the oceans. A benevolent bacterium. I called it plastophage. It was supposed to be my legacy. A gift to a planet choking on its own refuse. I'd been working on it for months, splicing genes from extremophiles, bacteria that thrive in harsh environments, into a common E. coli strain. The goal was simple: create something that could digest polyethylene and polypropylene without harming marine life. I sourced the base strains from online biotech suppliers. Nothing illegal, just standard lab fare. My setup was modest: a few incubators, a PCR machine I'd bought secondhand, and a CRISPR kit that made gene editing as easy as editing a document. I wasn't some mad scientist. I was a guy in his 40s, divorced, with a cat named Schrödinger who occasionally knocked over petri dishes. One rainy evening in October, everything changed. I'd been optimizing the plastophage's metabolic pathway, introducing a gene cluster from a deep-sea vent bacterium that enhanced its efficiency. The sequence looked perfect on my computer screen. Simulations showed it devouring microplastics at triple the rate of natural degraders. Excited, I prepared a culture in a nutrient broth, sealing it in a bioreactor flask. But in my haste, I overlooked a tiny detail: a contamination from an adjacent vial. It was a sample of a viral vector I'd been experimenting with separately, a harmless adenovirus modified for gene delivery. I must have cross-contaminated during pipetting. My hands were shaky from too much coffee. The next morning, I checked the flask. The culture had grown exponentially, a turbid swirl of life. Initial tests were promising. It chewed through a plastic film I'd submerged in the broth overnight. Elated, I scaled it up, transferring a sample to a larger fermenter. That's when the anomalies began. The bacteria weren't just eating plastic. They were replicating faster than expected, budding off viral particles that integrated into host cells with alarming efficiency. I ran a genome sequence on my old sequencer, and my heart sank. The viral vector had fused with the plastophage genome, creating a hybrid, a bacterium that could infect eukaryotic cells, hijacking them to produce more of itself while retaining its plastic-degrading enzymes. At first, I dismissed it as a quirk, cool side effect. I muttered, jotting notes. But as I tested it on simple eukaryotes, yeast cells, then algae, the implications dawned. This thing wasn't picky. It infected anything with a cell membrane, reprogramming it to prioritize plastic degradation. In a controlled environment, that was fine. But what if it escaped? Plastics are everywhere, in our clothes, our food packaging, our bodies via microplastics. If this microbe spread, it could turn the world into a degradation factory. I should have destroyed it then, incinerated the samples, bleached the lab. But curiosity got the better of me. I wanted to understand the fusion mechanism. Just one more experiment, I told myself. That night, I exposed a culture of human epithelial cells to a diluted sample. The infection rate was 90%. The cells didn't die, they thrived, but their metabolism shifted. They began excreting enzymes that broke down synthetic polymers. Harmless in a dish, but in a body, microplastics in human blood could be targeted, which sounded beneficial, until I realized the enzymes weren't specific. They attacked any carbon-based polymer, including those in medical devices, electronics, and infrastructure. Panic set in when I modeled the spread. Using basic epidemiology software, I simulated release scenarios. If even a milliliter escaped into the sewer system, it could hitch a ride on wastewater to rivers, then oceans. From there, aerosolization via waves, ingestion by marine life, up the food chain. Humans would get it through contaminated water or food. The hybrid's viral component made it airborne transmissible in high concentrations. Symptoms: initially mild, fatigue, as cells redirected energy to enzyme production. But as it accumulated, structural failures, pacemakers dissolving, four lines melting, even teeth fillings eroding if they contained polymers. The planetary impact was apocalyptic. Plastics underpin modern civilization, insulation in wires, components in vehicles, packaging for food preservation. If plastophage spread unchecked, power grids could fail as cable sheaths degraded. Transportation halts with tire and fuel line breakdowns. Food shortages from spoiled packaging. And ecologically, while it might clear ocean garbage patches, it would decimate plankton that rely on natural polymers, collapsing food webs. Coral reefs, already fragile, could dissolve if symbiotic algae were infected. The atmosphere might even suffer if it targeted atmospheric microplastics, altering cloud formation. I paced my lab, Schrödinger weaving between my legs. "What have I done?" I whispered. It was an accident, but intent didn't matter. This was a biological time bomb. I decided to contain it. I autoclaved most samples, but kept one vial for study, foolish pride. That night, a storm hit, knocking out power. The backup generator failed, and the fermenter's seal breached slightly. A droplet escaped, evaporating into the air vent. By morning, I felt off, itchy throat, mild fever. I tested myself, positive for the hybrid. It was in me. The infection was asymptomatic at first, but blood tests showed elevated enzyme levels. Microplastics in my system were breaking down, which was oddly satisfying, but I knew the risks. I isolated myself, contacting no one. But Schrödinger, he licked a spilled drop during cleanup. Cats have microplastics too, from their food. By evening, he was lethargic, his plastic litter box softening under his paws. I had to act. I emailed a colleague at the CDC anonymously, attaching genome data. Hypothetical bioweapon scenario, I lied. But the hybrid was already mutating in me, adapting. I felt sharper, strangely, perhaps a side effect of cellular optimization. Delusions? Maybe. I barricaded the house, but the vent led outside. Birds perched on the roof, if they inhaled spores. Days blurred. News reports trickled in, unusual equipment failures at a nearby recycling plant. Workers complained of rashes where plastic gloves touched skin. Coincidence? I doubted it. The hybrid was out, spreading via air currents, water, animals. I monitored online forums, biologists speculating about a plastic-eating plague. Governments dismissed it as hoax, but stock markets dipped on rumors of supply chain disruptions. Week 2, symptoms globalized. In Mumbai, slum dwellers reported water bottles disintegrating overnight. In Tokyo, subway cards melted in pockets. The hybrid infected fungi, accelerating decomposition in landfills, releasing methane bursts that exacerbated warming. Ocean buoys failed, hindering weather predictions. Hurricanes intensified without early warnings. I ventured out in a hazmat suit I'd improvised from non-polymer materials, cotton, glass. The streets were chaotic, cars stalled with degraded hoses, phones short-circuiting as casings weakened. People coughed, not from respiratory failure, but from inhaled enzymes irritating lungs. Hospitals overflowed, 4 bags leaked, prosthetics dissolved. The elderly suffered most, their hip replacements turning to sludge. The planet groaned. Amazon rainforests, already deforested, saw remaining plastics and research equipment fail, but worse, the hybrid infected soil microbes, altering nutrient cycles. Crop yields plummeted as plastic mulch degraded prematurely, exposing roots. Fisheries collapsed, nets dissolved, boats' hulls pitted. Birds dropped from skies, their ingested plastics metabolized into toxins. Governments mobilized. Quarantines, but too late. The UN declared a global emergency. Scientists raced for an antidote, using my leaked data. I revealed myself in a viral video, confessing the accident. I wanted to save the Earth, I said, voice hoarse. Instead, I cursed it. Hate mail flooded, some called me a terrorist, others a misguided hero. Month 1, civilization teetered. Power outages widespread as grid insulators failed. Internet flickered, fiber optics held, but plastic coatings didn't. Societies reverted, bartering food, using metal and wood. The hybrid evolved, targeting bioplastics next, then natural polymers like cellulose in plants. Forests withered, leaves breaking down prematurely. Carbon sinks failed, CO2 spiked. In my isolation, I experimented on myself. Injected antibiotics, antivirals, useless against the hybrid. CRISPR reversal. Too risky, it might accelerate mutations. Schrödinger died, his body a mushy heap. Grief fueled me. I cultured the hybrid from my blood, seeking weaknesses. Found one, extreme cold-halted replication. But how to deploy globally? A coalition formed, Russia, China, USA pooling resources, geoengineering proposals, seed clouds with antifreeze compounds, but the hybrid adapted, incorporating cold-resistant genes from infected polar bacteria. Antarctica reported ice core drills failing. Month 3, half the population infected. Mutations varied, some benign, granting resistance to pollutants, others lethal, dissolving internal tissues. Wars erupted over remaining resources. Borders meaningless as winds carried spores. Satellites fell silent. Plastic components degraded in orbit. I joined a resistance lab in the Rockies, airlifted under guard. We worked in glass enclosures, using pre-plastic era tools. Breakthrough, a phage virus that targeted the hybrid's fusion site. Tested on mice, success. Scaled production in bioreactors jury-rigged from steel. Deployment via drones. Spraying phage mist worldwide. Billions dosed. The hybrid retreated. Mutations slowing. Plastic stabilized. Though scarred, ecosystems rebounded slowly. Minus vast garbage accumulations now digested. Years later, the world healed, but changed. Plastics phased out. Biocompatible materials rose. I became a pariah. Then a cautionary tale in textbooks. My accident nearly ended us, but it forced evolution toward sustainability. The planet survived. Barely, but in quiet moments. I wonder, was it truly accidental? Or hubris disguised? The infection lingers in remote pockets. A reminder that one slip can unravel everything.